Professor at the Australian Research Centre in Complementary and Integrative Medicine, University of Technology, Sydney, Australia, and at the School of Medical and Life Sciences, Sunway University, Malaysia. With 14 years of teaching and research experience, he had completed a DST self-funded project on SNEDDS of polypeptide for diabetes mellitus. He had authored over 600 research papers with a cumulative impact exceeding 2,000 along with 5 books, 25 book chapters and 12 granted patents. His expertise lies in novel drug delivery systems, symbiotics-based formulations, analytical and bioanalytical method development and DAB studies. He has supervised 16 PhD and 35 master students and is currently guiding 6 PhD and 3 master students. Dr. Singh holds a H-index of 62 with 17,000 plus citations and is recognized amongst world's top 2% scientists by Stanford University in 2021, 22 and 23. He is the recipient of several prestigious awards including Dr. B.C. Dev Memorial Award for Popularization of Science, Bharat Siksha Ratna Award, Bharat Gaurav Puraskar and Distinguished Alumnus Award from Sri Siddhganga College of Pharmacy, Karnataka. Now, I request Dr. Sachin Singh Sir to share some insights on the topic Opportunities and Bottlenecks in Laboratory-Based Research Case Studies.
So, good afternoon everyone. Madam has given a very nice introduction of mine, but before going to the topic, I will give my simple introduction that I am an analyst. When I say this, it is important for all the audience that why I am saying, because if you see the research career of mine, many people talk to me that you are more than an analyst, you look like a formulation scientist. So I will tell the story and I will try to bridge the gap over there, that why it is like that and why bridging the gap is important, because it is very important for the young budding scientists like you people. And morning also, when our chief guest was there, Professor Dulal Panda, he was saying about that understanding of the topic is very, very important. And he had narrated also that analytical part on AI will become a boom in the future. So I will tell you, when I started my career on the M.Pharm, I did my M.Pharm from JSS College of Pharmacy, Ooty, and I was the gold medalist over there. And I have worked completely on bioequivalence and bioavailability studies on LCMS. Later on, I started working on dissolution science, and then I got to understand that why this bridge is important. And at the end of my talk, you will really see that why it is important, and I will give some of the answers also. So, when I have brought the topic opportunities and bottlenecks in laboratory-based research and case studies, I specifically seen the theme of the conference, and it was their challenges and opportunities. So, when you will go for this, it is a very simple thing, not made by me, it is given in the Google, okay, that if you want to execute any project, you need project design. So I will talk on this, that how you can formulate a hypothesis and plan an experiment with my own lab-based case studies. Okay, and second thing will be that experimentation and data collection, I will do that also. Because this is a sequence of the research, whoever is doing, whoever is sitting there, they used to follow the same. There is nothing new in this. But how actually we should reach there, I'll give you the case studies. I will tell why formulations are not in the market, so easily coming out, and what opportunity a lab student can get. But for that you have to be alert. Otherwise, whatever I will speak the best of the best, you will not get it. So you listen it. At least, you will understand that what is actually required. So after the data analysis and then communication and publication. Then opportunities are there. I will talk on that, but these are the highlights. That innovation at the bench level, highly fidelity of the data, collaborative environment and skill development. These are the 4 components which a student can get from the laboratory based research. But if you will go through that, the first part, this part, project design, you know whether the project will be culminated in a proper way, whether the project will reach at least to the research or patent or not, will be depending upon what type of project you are designing. 3 साल के बाद जब हम पेपर पब्लिश करने जाते हैं तब नहीं डिसाइड होता है, डिसाइड आपको डे 1 करना होता है. So then these were the opportunities. So I will about that, these are the constraints also, that resource and financial constraint. I will talk more on that. Target identification, validation, data management and technology transfer and scale up, which our respected VC sir was asking in the morning. So I will come to that. So for you people, I have brought one case study. I work in the area of diabetes. I work in the area of colorectal diseases. I do work on, as you have seen, BA studies. Most of my work is there, and now I am also working on neurodegenerative diseases. So when I say this, it means that an analyst is talking about the disease. It means that I start from ground zero, analytical method development, formulation, once again pharmacokinetic viability, and end up with pharmacodynamic studies. Entire circle of the point I used to do, and really I used to train the student in that way. None of the single master student in my lab is allowed to go without publication. And I will give you the examples also at the end of that, and within 6 months I am able to do it. But the journey is not so easy. I will show you. So I have brought for you polysaccharide based colon targeted delivery system. So when we talk, this is one study I have brought just to make you to understand the bottlenecks. What happens if we want, when we say that a delivery should be targeted, it means that the drug should not release at any point. It should only release to the target site. पूरी फार्मास्यूटिक्स जो है इसी पे काम करती है, नैनो पार्टिकल बनाए टारगेटिंग के लिए. But it's very tough to achieve target because nothing is in our hand. I'll show you. So why this colon targeting came? Because this work started in 1998, 1999 only. और आज कितना 2025, 25 साल तो हो गए. 25 years are done. Okay. Till now, not even a single colon target formulation is there in the market. Not even a single. जो इवोनिक फार्मा से निकल के भी आया यूट्राजिड, वो भी डिलेड रिलीज है. That's a delayed release formulation. That's not colon targeting. Delayed release means it will release after 2 hours. Means it will pass the stomach. और वो कौन सी है? मेसालामाइन की टैबलेट. People have tried with prodrug approach also. That was sulfasalazine tablet. That's why I have brought a very practical example of that. So these are the delivery systems which are used. Pressure control delivery systems, microflora, enzyme based, pH dependent system, time dependent system. Now here the challenge starts. As a topic is challenge and opportunities. So if you check out pressure control system, it hypothetically it is good. When it goes even to the in vitro data, pressure control system fails. Why? Because in our body peristaltic movement is there, and peristaltic movement is not constant always. Peristaltic movement changes in the body with respect to the type of environment, including your stress. So these type of delivery systems used to fail. Then comes pH dependent system. This is what Evonik Pharma and my friend Omji is sitting. He has worked in Coral Pharma and they have made lot of polymers for colon targeting. I hope so. But I am not criticizing, but none of the pH dependent system work. And that's why still we are having what I can say delayed release formulation. Why pH dependent system do not work? Because it has been tried a lot to make it colon targeting, and Evonik was the pioneer, in making the colon targeted formulations. But what happened that they have researched and they brought Udrazid FS30D. They have brought S100 and they have brought Udrazid RS also. Three type of polymers they have developed. And after that lot of polymer making company also tried कि colon targeting के लिए लाएं ताकि colorectal cancer को ठीक कर सके. Colorectal cancer. So that at least targeting will be there, so patient will be having no issue, because you all know that cancer medicines are very costly. So why not we can make the people to reach to that side that even a poor person can also get the system. So the best affordable system which came into the market was a pH dependent system, but it failed. You know why? When we talk about the pH of the GIT, anyone knows what is the total pH of the GIT? Anyone? What is the pH of colon? Exactly, 7.5. 7.5 is the pH of the colon, and GIT pH, gastric pH is 1.2. After that it follows to intestine, small intestine, then it goes to large intestine. When the pH travels from stomach to intestine, 1.2 to 6.8 shift happens. This is very important. What happens that when the student used to go for the literature, I have seen even in my lab, they will go to PubMed, they will just look at their abstract. And after looking up the abstract, whatever they want, that data they will take and that's all, ho gaya. They will not read the entire paper. So you have to read the entire paper, you have to go with the patent search data, you have to go with the newspapers. You will understand that really you are working on a thematic area or not. Really your problem will be able to solve the issue or not. Means your research will be able to solve the issue or not. So I am saying you because we have worked a lot and I have seen that pH delivery system you don't use to work. You know why? Because when we will be having the disease condition, most of the time pH becomes alkaline of the entire body. A person suffering from ulcerative colitis, a person suffering from colorectal cancer, वो already पीड़ा में होता है. The person is having a pain. But a person is suffering from ulcerative colitis or colorectal cancer, they will be having alkaline pH in the body. FS30D they have brought for making colon targeting, and FS30D was also about to release at 7.5 pH. So what happened that when the clinical trial has happened, it was good on healthy volunteers. It was good on healthy volunteers because the way they triggered the release it came. But do we really give medicine to healthy person? Do we give medicine to the healthy person? Whom we give? Patient, diseased person. So after 2 hours itself, the formulation was released. So finally what they have decided that HPMC लो या तो sustained release बना दो या delayed release बना दो. इसके ऊपर पॉलीमर कंपनी ने काम नहीं किया. This is fact. और आज भी आप मार्केट में देख लो, आपको मेसलमाइन की टैबलेट जो मिलेगी, इवन 5FU को भी लोगों ने ट्राई किया, दे विल बी डिलेड रिलीज और सस्टेन्ड रिलीज. पॉलीमर की कंसंट्रेशन बढ़ा दो, बट प्रॉब्लम वहीं रह गया. टारगेटेड मीन्स 90% ड्रग शुड रिलीज इन द कोलोन ओनली, नॉट बिफोर दैट. सो इन दैट केस pH has also gone. So ultimate thing was the enzyme-based prodrug approach. The friends who are sitting from chemistry department, they know that enzyme, and the students also, that it is a lock and key hypothesis. In body, when it goes in the GIT, we are not having one enzyme, we are having multiple enzymes. So making an enzyme trigger system is also very tough, because we will not get a specific enzyme for a specific polysaccharide और स्पेसिफिक ड्रग. आपको एंजाइम चाहिए तो आपको प्रोड्रग बनाना होगा, और हर ड्रग की प्रोड्रग नहीं बन सकती. So what we talk in the research will reach to the bedside of patient is not always true. Research is something, making it to translational. Even morning also, Professor Panda was saying about that TLR4 तक ही पहुंच पाते हैं. We used to reach up to TLR4 only. TLR5 में academic institutions नहीं पहुंच पाते हैं. I hope you all were there in the session, right? So we started working on microflora activated system, and this was not our idea that I should work or we should work on microflora activated system. I'll show you. So this already I said. In this, if you see the PPT, it is written how a polysaccharide based delivery system works. Okay, ये light नहीं आ रही है properly. Okay, so development हुआ, oral delivery हुई. So the formulation is that polysaccharide can only be digested by the bacteria which is present in colon. बीमारी होगी तो भी बैक्टीरिया मिलेगा, नहीं होगी तो भी बैक्टीरिया मिलेगा. That was the thing. So based on that, this is why this system became very good, that it can work and it can give targeting to the colon. That is called microflora based system. So in that what happens, that when a drug reaches, if you see in the figure, it is clearly written that once the drug is there, the blue color is the polymer coat. The blue color is the polymer coat. It only gets dissolved. Here it is there. It only gets dissolved when it will get the microflora. So it does not require any pressure control. It does not require any pH. It will work because you also know that polysaccharides can only be digested in the colon because they have lot of microbes in that. So we got opportunity now to work out. We got opportunity to work out that हाँ यहाँ से कहानी शुरू करनी है क्योंकि हमें gap मिल गया. We got the research gap. Then we started doing the research. So I have brought some of the paper which I referred when I started working on this. So it was Dr. Y.S.R. Krishnaiah lab in Andhra University. They have worked a lot on the polysaccharide based delivery system. If you will see the year, you can find that it is in 1999 and 2000. We started working in 2011. 1999 से research चल रहा है. 2002 आपको मिलेंगे 99 के भी papers. So what is there that they started working on polysaccharide based delivery system. ये हमारा discovery नहीं है. So we started doing the research. Then if you see the papers, they are having lot of work on that. They tried with different drugs. With research ऐसे ही होता है. छोटे एक बार जब ट्रिक मिल जाती है तो हमें बहुत सारे ड्रग पे ट्राई करना होता है कि वाकई में ये डिलीवरी सिस्टम काम करेगी कि नहीं करेगी. तो इसको बोलते हैं अपॉर्चुनिटी. हाउ यू कैन डिजाइन एंड टेलर योर वर्क. Then if you see this 5-fluorouracil which has been gone up to phase 3 of the clinical trial by Professor V.R. Sinha lab from Punjab University, Chandigarh. And this was a collaborative work of Naipar Mohali, the first one. This was a collaborative work of PGIMR, आप जानते होंगे, Post Graduate Institute of Medical Science, Chandigarh. And it was a collaborative work of Punjab University. 3 topmost universities working in the area of pharmaceutical sciences and medical sciences started working, and PGI इसको phase 3 तक ले गया. The concept started by YSR कृष्णैया. देखो मेरी अंडरस्टैंडिंग जो सुबह में बोल रहे थे पांडा सर, अंडरस्टैंडिंग रिसर्च करवाना है तो अपनी अंडरस्टैंडिंग मुझे पहले डेवलप करनी, बिकॉज आई एम ए टीचर, देन ओनली आई कैन ट्रांसफर टू द स्टूडेंट्स. So my understanding is more important than the student understanding, and then only student understanding can be given in depth. So what happened? We started from here, but this also failed in phase 3 clinical trials. Why it failed? I will show you. The actual problem was this: when the formulation was once again given to the healthy patients, it worked. That's what is the first diagram one may have taken. That was the first diagram. And then the second diagram says that when the person will be a patient, drastically the microflora count decreases because of gut dysbiosis. आप पढ़े होंगे, आजकल गट पे बहुत काम हो रहा है. Lot of people are working that gut microbiota, gut brain axis, gut. If gut is good, everything is good. So here the challenge came with this delivery system. और मैं बिल्कुल ट्रांसलेशनल पार्ट बात कर रहा हूं. The challenge came here that when the concept was, when the drug will go, bacteria will eat the polysaccharide, right? But a person suffering from ulcerative colitis or colorectal cancer, you'll see the literature, because of gut dysbiosis, the bacterial count, particularly gram-positive bacteria count, goes very down. So what happens once again? The bacterial count gone as such. If you see, drug is coming out of the colon as such. मतलब drug release ही नहीं होगी. And here only the polysaccharide based delivery system failed. This is literature talking, I am not talking. So then the question was there that how then we will go further. Then 2011 I joined the lab, okay, and we started working on that. Then came the third part. Now you see it very clearly. Extra probiotic supplementation to a diseased patient. बेटे जो पीछे बात कर रहे हैं, either you listen or you can go. So what happened here? That probiotic, when we will supplement the probiotic along with the polysaccharide based delivery system, in the first dose nothing is going to change, because gut microflora will also start generating and it will take some time. So but once you are giving continuously co-administration of a probiotic based delivery system, so what will happen? That polysaccharide, when it will reach, because the is getting compensated, the bacteria will eat the delivery system and drug will release to the colon. And here we found that the gap can be overcome by using additional use of probiotics. अब रिसर्च शुरू हुई. Now what happened? So we prepared first mesalamine microspheres. I'll not go to the scientific part deep into that, because my objective is the opportunity and challenges, not the nanotechnology microsphere. For that I can have a different platform. Okay, so we prepared microsphere, in which microsphere was prepared with guar gum and xanthan gum, but in the same capsule we have administered probiotic also. So this was a new concept. This was a very new concept. And what happened next? So we got some result. We tried with glacial acetic acid induced colitis model also. and then we got good result, promising result, and after getting the promising result, we published it. Not only we published it, the data, but we went for getting the filing of US patent, China patent, Indian patent. The patent has already granted to us. Okay, so we went for filing that. And after filing, what we did? Now the story started, next story, next challenge. When we got this technology in 2011 and 2012 after publication, what happened? We started moving towards the tech transfer. In Dehradun, you have 50 lakh initial seed money to do the know-how to the company. अब सर आपके सवाल का जवाब आएगा. Before going to this, when we started doing the know-how, company said that we will not do this MOU. In money they gave us 3.5 lakh, and after that they said that हम नहीं करेंगे. You know why? Research is something very good to do. We got a very nice publication also, but when it went to industry is having different objective. पहले तो इंडस्ट्री वालों ने हमें ये बोला कि नो 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 नो, वी हैव टू डू इट बिकॉज दे आर वेरी प्रैक्टिकल आल्सो, दैट इफ यू आर डूइंग इट, इफ यू आर गेटिंग 1 ग्राम ऑफ माइक्रोस्फीयर, अगर जो फॉर्मुलेशन के साइंटिस्ट है दे नो इट दैट मेकिंग माइक्रोस्फीयर इट टेक्स टाइम एंड द यील्ड इज वेरी लेस. तो नोवेल्टी तो थी, काम में नोवेल्टी तो थी, इसलिए पेटेंट भी आ गया, पेपर भी पब्लिश हो गया. बट पेटेंट टेक ट्रांसफर के लिए जाने में चैलेंज आ गया, बिकॉज दे से दैट if 1 gram of mesalamine microsphere we will get in 2 days after costing too much, our cost is going very high. हम सैलरी भी कैसे निकालेंगे? This is now the challenge. After that we went with our entire thing to the Dehradun to show them how to work. उसके बाद इंडस्ट्री के दूसरे डिमांड आए हमारे सामने, जो हमारी इंडियन इंडस्ट्री की सबसे बड़ी प्रॉब्लम है. दे सेड दैट आप टैबलेट लाओ जी, हमारे यहाँ तो टैबलेट बनते हैं. सर दिस इज द बिगेस्ट प्रॉब्लम ऑफ फार्मा इंडस्ट्री. वो जो बनाते हैं वैसा ही चाहिए, नया सेटअप नहीं लगाना चाहते. R&D वाले पीछे हट जाते हैं. इंडिया में R&D है ही नहीं, डॉक्टर रेड्डी ने अपना क्लोज कर दिया. अब जो R&D है वो कॉस्मेटिकल्स में ज़्यादा है. दिस इज फैक्ट डियर स्टूडेंट्स. सो वी स्टार्टेड वर्किंग कि अब क्या किया जाए भाई, कैसे लाया जाए, क्या किया जाए कि ये टैबलेट में, क्योंकि देखो ये पॉलीसैकेराइड है एंड इट इज नॉट इजी दैट यू कैन मेंटेन द कंप्रेशन फोर्स बिकॉज प्रोबायोटिक विल डाई. प्रोबायोटिक कंटेन्स लाइफलाइज फॉर्म ऑफ बैक्टीरिया, सो इफ यू गिव ए वेरी हार्ड कंप्रेशन फोर्स बैक्टीरिया विल डाई. तो कॉन्सेप्ट तो वैसे भी फेल हो जाएगा, राइट? कॉन्सेप्ट भी फेल. सो वी गेव दिस टॉपिक टू अनदर एम्फार्मा स्टूडेंट. तो चलो अब कुछ और करते हैं. So Prithviraj, right now he is leading the Omics Pharma in Hyderabad. So we gave to Prithviraj, and we did it in collaboration with JSS Suti because right that time we were not having a student. Now we are having very good external manager. आपके यहीं का है, उमंग फार्मा का ही है हमारे पास. So he went to the JSS and he prepared pellets कि चलो कुछ तो कंपनी को दें. कंपनी को वो नहीं चाहिए तो चलो पेलेट्स बना के पेलेट्स के साथ हम देते हैं. इजिली वी कैन फिल इट इन कैप्सूल. तो बेस्ड ऑन दैट वी डिड दिस रिसर्च. अब देखो चैलेंज था बच्चे, आपके लिए यही जरूरी है समझना कि वॉट एवर द कंपनी एंड इट्स नॉट इजी टू ब्रिंग द प्रोडक्ट, बट यहां से अपॉर्चुनिटी खोलना इंपॉर्टेंट है. how next opportunity will come, how we can go with the research, so the research should not die and next opportunity will come. So we went with this. We made sulfasalazine. We tried with a model drug sulfasalazine to make pellets. We coated with guar gum, but इसमें भी challenge आ गया. After that we published this paper also in a very reputed journal. But once again problem came. You know what? 1 gram of my friend Dr. Om ji was saying that a small material he is having अभी भी है इनके पास बैग में कि इसको सचिन जी इतना लोगे तो इतना बन जाएगा. 10 रुपए की चीज आएगी और 1000 में बिकेगी मार्केट में. बिकॉज ही इज ए पॉलीमर साइंटिस्ट. सर दे भी रहे शेयर भी ही इज ए पॉलीमर साइंटिस्ट. ऐसी क्या होता है गुअर गम, ये जो पॉलीसेकेराइड होते हैं ना, 1 ग्राम फूल के 1 बाल्टी हो जाएंगे. If you add guar gum, it will keep on swelling, swelling and swelling. तो क्या होगा? पीछे जाते हैं. ये सेकेंड पार्ट में दिख रहा है कोटेड स्टेरॉइड, ये नहीं आएगा. It can be done on the laboratory scale. Laboratory scale पे तो हमने बना लिया. But when it will go to scale up, practically we have seen सारा agglomerate होने लगा, चिपकने लगा एक दूसरे से. Because when you will do the aqueous solution of guar gum, it will swell one thing, and evaporating water is not easy from the coating. And if you will not evaporate properly, it is containing guar gum, it will have the fungal growth, bacterial growth शुरू हो जाएंगे. तो ये सब चैलेंज भी हैं, बोटलनेक्स हैं. आप जब फॉर्मुलेशन बनाते हो तो, और कंपनी को सब कुछ करते हो, आपको स्टेबल फॉर्मुलेशन देना है, कॉमर्शियल फॉर्मुलेशन देना है. द अदर प्रॉब्लम व्हाट वी आर फेसिंग विद दिस, आई विल शो यू. बड़ा वर्सटाइल सा काम लगेगा मेरा आपको कि कुछ अलग अलग डायमेंशन में मेरा काम जा रहा है. चलो ठीक है, जैसा भी जा रहा है, रोलर कोस्टर होना चाहिए, राइट. सो वी केम टू नो दैट डिसोल्यूशन टेस्टिंग ऑफ पॉलीसैकेराइड बेस्ड फॉर्मुलेशन इज आल्सो नॉट इजी. अब उसमें क्या चैलेंज है मैं दिखाता हूं. The available techniques which were there for the polysaccharide based delivery system, enzyme based delivery system, already I said, अगर आप read करोगे, कर लेना, it is enzyme and polysaccharide specific. So whenever we are changing the polysaccharide and all, what will happen? We have to give a specific enzymes. It is not possible always. And in body, as I said, gut microbiota contains number of polysaccharide, a number of enzymes. All the enzymes cannot be added in a single dissolution medium. So how we will get in vitro in vivo correlation? IV IVC कैसे होगा? The second one was there, which were most popular. सबसे popular है rat cecal contents. So when we are also doing the practical for our M farms, we used to sacrifice the rat. उसके लिए हम CPC से परमिशन लेते थे, and then we used to sacrifice the animals, and then we used to take out the cecal content. अब सोचो, एक बार formulation बनाया, once only we made the formulation, and we have to use cecal content, at least we have to sacrifice one rat. Formulation will not be possible always in the first step itself. We need number of trials. And think when it will go to the industry, every day quality control will be there, isn't it? Every day for batch release quality control is required or not? बिना QC के कोई batch release होती है? So for every QC, do we need to sacrifice rat cecal and take out the rat cecal content? ये दूसरा चैलेंज आया हमारे पास कि भैया फॉर्मुलेशन ही नहीं बनानी है, उसका मीडिया भी चैलेंजिंग है, उसका QC भी चैलेंजिंग होगा. फिर मैं तो एनालिस्ट था, आई वाज एन एनालिस्ट, राइट. ह्यूमन फीकल स्लरी कोई उठाने के लिए नहीं तैयार है और इसके लिए एक्सपर्टीज चाहिए. Human fecal slurry, we need the donors, healthy donors, and the donor should not have taken antibiotics from last 15 days, नहीं तो उनका माइक्रोफ्लोरा मर जाएगा. These are the bottlenecks with the delivery system. एक छोटी सी चीज नहीं है इंडस्ट्री में. इंडस्ट्री को अगर कोई चीज देनी है तो आपको परफेक्ट देना होगा, तब हम TLR 2 से 3 तक जा पाएंगे. But where challenge is there opportunity. In this paper we have given all our challenges. What are the challenges are there? Everything is given. If you refer this paper, you will understand. We have discussed all the challenges with the police secretary delivery system. Then I sat with my students and I thought कैसा ऐसा एनारोबिक कंडीशन किया जाए शरीर के बाहर कि एक मीडिया तैयार हो जाए. What should be done? So फिर बेसिक्स में गए, इंडियन फार्माकोपिया देखना शुरू किया. Then I got one medium, fluid thioglycolate medium, आपने पढ़ा होगा, for anaerobic culture. तो वहां से दिमाग में एनालिसिस तो था ही दिमाग में, because I was an analyst. So from there we started thinking, can we do a culture where bacterial fermentation can be done outside? अब दही से भी आप कर सकते हो. You can use curd also. यहाँ तो श्रीखंड काफी ज्यादा खाते हैं गुजरात में, राइट? वो भी फर्मेंटेड प्रोडक्ट है, उसमें बैक्टीरिया बहुत होता है. वेरी हेल्दी फूड इज श्रीखंड, बिकॉज जो फर्मेंटेड है. बट दैट इज नॉट फिजिबल एज सच. आप नहीं उसको टेक ट्रांसफर कर सकते. सो वी डेवलप्ड अ मीडिया, वेरी सिंपल मीडिया. प्रोबायोटिक को लिया, हनी बेस्ड भी मीडियम बनाया था हमने. फ्लूइड थायोग्लाइकोलेट मीडियम में हमने मीडिया तैयार कर दिया जी. वी मेड इन विट्रो मीडियम एंड वी हैव कंपेयर्ड इट डेटा. वही यहां दिखाया गया है कि हाउ द डेटा इज कमिंग आउट कंपेयर्ड, क्योंकि आप वैलिडेट करना जरूरी है उसको. जो भी बनेगा, जो भी सिस्टम बनेगा, उसको वैलिडेट करना बहुत जरूरी है. सो वी वैलिडेटेड विथ फीकल कंटेंट एज वेल एज ह्यूमन फीकल स्लरी, बिकॉज यही सबसे ज्यादा बायो रेलेवेंट है. तो we validated and we found synchronized result. So immediately when we got result, as a scientist, it was our duty to immediately publish it. It is the only paper available in the entire world on this concept in our lab. And then what happened? We got a patent granted immediately of this medium. देखो कैसे अपॉर्चुनिटी निकल रही है. गुजरात में हर चीज को बहुत अच्छा जानते लोग बिजनेस करना. ऐसे एक टीचर और एक रिसर्चर का काम है कि उसके लैब में एक छोटा सा भी केमिकल अगर बच्चे ने छू दिया तो पब्लिश होना चाहिए. होना ही चाहिए. होना चाहिए, नहीं होना चाहिए? यस. इसलिए मैंने बोला आप जैसे जैसे मेरे स्लाइड में बढ़ते जाओगे आपको इंटरेस्ट भी आएगा और समझ भी आएगा कि आगे से आपको क्या करना है. यू हैव टू बी फोकस्ड, दैट्स ऑल. रिसर्च गैप ढूंढना इंपॉर्टेंट है. क्या लगा? कुछ नहीं लगा. रैट सीकल कंटेंट के लिए सीपीसीए का परमिशन लेना होता था, सब कुछ लेना होता था, राइट? और कॉस्टली भी था, एनिमल सैक्रिफाइस भी था. 5 मिनट में फ्लूइड थायोग्लाइकोलेट मीडियम लिया, प्रोबायोटिक डाला, कार्बन डाइऑक्साइड रिच एनवायरनमेंट बनाया मोमबत्ती से. वो भी कोई सप्लाई भी नहीं किया है, हमारे पास सप्लायर, लेकिन नहीं किया. और 20 रुपए में मीडियम तैयार हो गया. ये तो साइंस है. 20 रुपए में मीडियम तैयार हो गया. लैबोरेटरी को एक नया मीडियम मिल गया, स्टूडेंट्स को एक नया पेपर मिल गया, यूनिवर्सिटी को एक पेटेंट मिल गया. सारा काम हो गया. So this is how challenges become opportunity. अगर एक चैलेंज नहीं समझते हम प्रॉपरली, लिटरेचर बिल्कुल सही से नहीं करते, how would have reached the solution? But story is not ending here. अभी भी पुराना चैलेंज है. कोलोन टारगेटिंग चाहिए कंपनी को. और कैसे चाहिए सर? टैबलेट चाहिए उनको. क्योंकि हमारे यहाँ के जो मैन्युफैक्चरर हैं वो टेबलेट और कैप्सूल ही समझते हैं, इससे ऊपर कोई इन्वेस्ट करने के लिए तैयार नहीं है। जब अभी हम क्लोज मीटिंग में भी बैठे थे बीसी सर के साथ, तो दुलाल पांडा सर वाज सेइंग दिस ओनली दैट इंडस्ट्री इज नॉट रेडी टू कम अप। 20 लाख के फंड में क्या होता है? 20 करोड़ 200 करोड़ चाहिए इंक्यूबेशन सेल को, अगर एकेडमिक रिसर्च को पहुंचना है वहां तक। तो हमने काफी, ये इजी लग रही क्योंकि मैं बोल रहा हूं, मैं इसलिए बोल रहा हूं क्योंकि मैंने अपने जूते रगड़े हैं। इंडस्ट्री को कन्विंस करना आसान नहीं है। सो बना दिया जी मिनी टैबलेट। सो दिस वी गेव टू वन टॉपिक टू आवर पीएचडी स्कॉलर। ओके, अब चलो मिनी टैबलेट बनाना। मिनी टैबलेट इमीडिएट रिलीज वाला नहीं चाहिए, कोलोन टारगेटेड चाहिए। मिनी टैबलेट तो बहुत बनाते हैं कंपनी, बट those all mini tablets are fast dissolving or immediate release. Those mini tablets are not colon targeted tablets. मिनी टैबलेट्स तो है मार्केट में, पहले से थे, तभी हमने भी बनाया, बट those mini tablets are not colon targeted tablet। हाथ में लेते ही टूट जाते हैं। मिनी टैबलेट को हम हाई कंप्रेशन नहीं दे सकते, बट यही हमारा रिसर्च था, क्योंकि बैक्टीरिया को हम कंप्रेस नहीं कर सकते, राइट? आई सेड दैट बैक्टीरिया वी कैन नॉट कंप्रेस बिकॉज दे आर डाई। प्रोबायोटिक इफ वी कंप्रेस स्ट्रॉन्गर, 10 किलो न्यूटन तो कम मिनिमम कंप्रेशन फोर्स होता है। अब यहां पे देखो, ये भी बोटल नेक है, टैबलेट भी बनाना, बैक्टीरिया भी नहीं मरना चाहिए। सो व्हाट वी डिड, वी मेड द मिनी टैबलेट and we started coating over it. We did some alteration in our coating composition also, okay, and then because it requires very low compression force, we added in the blend itself probiotic, and we made with a very less compression for this tablet, and then we coated that. And then we did all these studies. You can see pharmacokinetic study to prove that, cell line studies also, in Kakutu cell lines we have done, and then we reached up to here. So still we gave it to the company, और वहां से कंपनी ने अपना स्टोरी बदल दिया। जब उनके हाथ में गई चीजें, तो कंपनी बोलती जी अभी हम ये प्रोजेक्ट नहीं कर रहे हैं, अभी हम दूसरा प्रोजेक्ट कर रहे हैं, क्योंकि हमने उनको बोल दिया क्लिनिकल ट्रायल शुरू करवाओ। एनिमल पे तो हमने करके दे ही दिया। अब नेक्स्ट चैलेंज था सर उनसे फंड निकालना कंपनी से, वहां वो बैक स्टेप कर गए, बिकॉज दे आर नॉट रेडी टू टेक इट फॉर द क्लिनिकल ट्रायल, क्योंकि उनको वहां पैसा लग रहा है। बिकॉज वो मेरा काम नहीं है, मैं एकेडमिशियन हूं, मेरी यूनिवर्सिटी का भी काम नहीं। वी नीड फंड्स, एंड व्हाई वी वेंट टू द इंडस्ट्री, बिकॉज दे वर हैविंग DSIR अप्रूवल। क्लिनिकल ट्रायल इज ऑफ कोर्स a very costly process which got lot of fund. So from here the journey has not ended, lot of works are going on, we are publishing continuously, okay, and we are working in multi-dimensional areas, as I said। एक दो बच्चा कोलोन पे करता है, कोई न्यूरोडिजेनरेटिव पे करता है, लेकिन एक चीज कंपल्सरी कि हर बच्चा whoever comes in my lab, they used to work completely, they do the analytical method development followed by formulation development followed by bioanalytical development and then pharmacokinetic and pharmacodynamic study। लैब में आया है तो फॉर्मुलेशन, एनालिटिकल और एनिमल हैंडलिंग सीख के ही जाएगा, वरना मेरे पास नहीं आएगा। क्लियर हो? पाली बच्चे को बोल दिया जाता है, यू हैव टू वर्क नाइसली, पोलाइटली, 9 to 5 इज सफिशिएंट। 8 घंटे, मेरे गाइड बोलते थे 8 घंटा बहुत होता है। इफ एनी स्टूडेंट इज वर्किंग 8 hours एवरी डे इज सफिशिएंट। कोई रुक के देर रात काम करने की जरूरत हमेशा नहीं है, किसी 1-2 दिन पड़ती है, बट उसमें तो सिंसियरिटी होनी चाहिए आपके 9 से 5 में। सो एनीवे, सो देन वी मूव्ड आवर रिसर्च टुवर्ड्स द फीकल माइक्रोबायोटा, कि अब कुछ इसमें भी किया जाए। फीकल माइक्रोबायोटा मतलब ह्यूमन फीकल लेके आज के डेट में ट्रेंडिंग टॉपिक है। फीकल माइक्रोबायोटा ट्रांसप्लांटेशन इज ए ट्रेंडिंग टॉपिक नाउ डेज। अब इसमें PGI में इस पे ट्रायल्स भी चल रहे हैं पर क्लोस्ट्रीडियम डिफिशियल इन्फेक्शन। अब यहां पे भी बहुत सारे चैलेंजेस हैं। फीकल माइक्रोबायोटा, इफ आई विल से यू दैट दिस इज ए फीकल माइक्रोबायोटा, विल यू ड्रिंक? When we will say that this is a fecal microbiota, fecal sample, fecal की slurry, anyone will drink? Yes or no? No. Because सुनते ही नाक चढ़ने लगेगा, right? We made self nano-emulsifying drug delivery system, okay, using a mixture of curcumin and fecal microbiota, where we have done the solidification. And now those who are the formulation scientists, they know that SNETs require for the delivery water, rather than using water we have used fecal slurries, क्योंकि जब नेजो से देते हैं ना कैंसर के पेशेंट का डिफिशियल में, तो उनको वोमिटिंग हो जाती है, रिफ्लक्स हो जाता है, दे कैन नॉट टेक इट। सो व्हाट वी डिड, कुछ ऐसा किया जाए कि वो सैंपल हाथ में भी आ जाए। और मैं एक छोटी स्टडी दिखा रहा हूं, हमने मेटाजीनोमिक सब किया हुआ है उसके लिए। We have done all metagenomic study and all also, कि ताकि कोई कल को ये ना बोले कि भाई पहले आपका माइक्रोबायोटा था, प्रोसेसिंग में मर तो नहीं गया। तो देखो, पब्लिश करने के लिए और चीजें करने के लिए सारा डाटा होना चाहिए आपके पास, अदरवाइज कोई फायदा नहीं है। So we applied QBD because I'm also an QBD expert. So I used to talk a number of times on formulation or analytical method using quality by design. So any of my formulation utilizes QBD. Without QBD I don't go. Okay, so we did QBD and then we did the fecal microbiota, and all are ethical approved. All what I am showing, none of them we have done as such. We have taken earlier ethical clearance and then only we have done it. So we made fecal microbiota sample. We have converted into the powder. अगर मैं किसी को दे दूं बिना बताए कि fecal microbiota आराम से खा लेगा. And come to my lab, which flavor powder you want, it is there with me. Vanilla flavor, pineapple flavor, chocolate flavor. If I will not say, curcumin itself is a wonderful, it comes out very nice golden yellow color. When we solidify, when we lyophilize it, it comes out with a very golden yellow color. But देखो novelty है चीजों में, कुछ अलग है, इसलिए पब्लिश होगा, इसलिए पेटेंट भी होगा. तो आपको रिसर्च गैप ढूंढना ही पड़ेगा. So if you are investing your time for doing the research work, try to find out the failures of previous papers, and from there you make your opportunity in the laboratory based research. And when you are doing a lab research, you should be always aware that what is the capacity of my lab. I always say to my friends and students that try to see the strength of your lab. अपने लैब में क्या फैसिलिटी, उसके अंदर काम करो. आइडिया वैसा जनरेट करो. You have to do the idea generation in such a way that you can do the work in your own lab. Otherwise क्या होगा आपका, आप कभी एक उस लैब में जा रहे हो वर्क करने के लिए, किसी दूसरे कॉलेज में जा रहे हो, कहीं से मंगवा रहे हो थर्ड पार्टी के डाटा का क्या भरोसा है. थर्ड पार्टी डाटा क्या दे रही है आपको, एंड दैट विल किल द टाइम. 6 महीने में एक एम-फार्म के बच्चे को काम करवाना है, और आई विल गिव द हाइपोथेसिस, सच ए बिग हाइपोथेसिस, देखो बच्चा के एपीआई या लैब में घूमते रहे तो उसका तो काम होगा ही नहीं, वो क्या ही पब्लिकेशन करेगा, नहीं कर सकता. He can't do, because half of the time has gone in searching the lab where the formulation can be feasible, where the analysis for analysis, if you have to go every day you need HPLC, every day you need UV to do that. So what happened, so we did all the studies, and then we have done for PCOD also, clone gut and that axis, okay, and then we have done in PCOD model for this work, and then we published it also in a very reputed journal of material science, Material Today Chemistry. We published it, and then We have the application filed also. It is under, we have filed a request for examination, and soon I am expecting that this will get granted. And then we had, because I have given you the gut microbiome concept, so we have taken with another topic to the diabetes also, where we have taken mushroom polysaccharide and probiotic, and we did the diabetes studies. Here also number of different with a small, you won't believe, with a petri plate and a glass rod, some formulation can be made. Snakes is one formulation which you can make in one day. That's why it's scalable. A company is there in Kerala from which, because it comes under nutraceutical, a company is there in Kerala from which our talk is going on for tech transfer. This work has got PCT filed also. We have filed Indian patent, we have filed PCT also. PCT, those who know, they know that your data will be protected for 140 countries in the world. So we have done it, dissolution profile. दोपहर का टाइम है, मैं ज़्यादा डीप नहीं ले जाऊंगा आपको. Okay? Simple, क्योंकि डाटा आप ऐसे नहीं समझ पाओगे, because I have to show you, the expert will understand this data. The senior professors who are sitting, they will understand that this is the religious study. So we have got some significant results. Okay, we have done complete kinetics, histopathology, all the things we have got. Okay, and then we have done the PCT filing also, and simple with this concept, polysaccharide probiotic based concept, that इससे हमने different formulations बनाए. Not only the snakes we made liquid solid powder technology also. We published in a liquid form also. We have done a spray drying also. Then we made pellets, spirites. We published that also. Okay, so with one work, one concept, see 4 or 5 different type of technology we developed. We published it also in some good quality journals. And then, as I said, we have done the PCT filing also, which is also known as world patent, because it is there in 140 country, protection is there. Okay, and this is my fresh students team who is working on, who has just passed in the few years. I hope Nikhil is also there. Nikhil, where is Nikhil? Nikhil, can you stand? Nikhil is there, one of the products of my lab. You can see Nikhil is there also somewhere. Yes, hai Nikhil. Thank you, Nikhil. Sit down. And I'm here speaking also because of Nikhil and his team, who has worked continuously to make this conference to happen. And currently, Dr. Sukriti has passed out these 2 and rest all are my PhD scholars who are working right now with me. And most of them are working on the neurodegeneration, a few are working on the other models also. And then my important thing is collaborations also. So I like to acknowledge my collaborators also who are working with me. Across the globe, I am having collaborations. Few I have mentioned here. So these are my collaborators also. So drug delivery, if I have to consult, I consult with Professor Patrawale, a renowned personality in India. Professor D'Souza is also there, who is leading in IPA also, I hope in Maharashtra, sir might be knowing. And then my own team is there. Dr. Kamal Dua is one of the renowned scientists in lung biology. So I used to collaborate with Kamal also, he is my very good friend also. Professor Gautam Rajan is there, he is a design expert also. I hope Kamal lecture is also there tomorrow. You listen him, you will be overwhelmed, his lecture. So Professor Gautam Rajan, who is my boss also, so he is there with QBD expert also, and he is working on space research right now in JSS. And Neil Davis and Reimer, they help me in analyzing the data of a statistical point of view in pharmacokinetics and biostats. And Flavia is there if I am doing some gold nanoparticle synthesis. So Flavia helps me in green chemistry. Professor Teresina is also there, who helps in biochemical analysis and all. Go is there, who works in medical devices and biomedical sciences also. So they all are, whom I have shown, are very, so both close collaborators, and they are my very close collaborator. More than collaborators, they are my friends. So this is there. So I hope you understand that why because many times this question comes in pharmacy department that ये pharmaceutics है, वो ecology है, वो chemistry है, वो analysis है, वो QS है, वो regulatory है. See, if you have to work on yourself. God has given us two upper limbs, two lower limbs. If one of the limb will also go, what will happen? Little challenge will come in quality of life, right? Yes, this is my last message to you people. So, if you have to work in lab and you have to supervise your students, so you should know formulation also. You should know analytical things also. You should know pharmacology. So मैं खुद से जाके जानवर को हैंडल करके सिखाता हूँ बच्चों को. अब थोड़ा कम हो गया because I am heading the department also. So मैं खुद से जाता हूँ सीखता हूँ क्योंकि पहले मैं सीखा हूँ काम को और उसके बाद सिखा रहा हूँ. ये 2011 से 17 तक मैंने सीखा ही है बच्चों के साथ लग के. So the journey goes like this. Start analytical work, do the formulation development. और मुझे आना चाहिए. I cannot expect that my student should go to the other supervisor and should ask. ये मेरा failure होगा. So that's why I learned and I train my student in that way. So these are the two people who have made these slides, Dr. Sukriti and Kosto. Dr. Sukriti is currently working as Associate Professor at Chitkara University. Time to acknowledge your family who has given me this platform to share. At least 1 hour I have taken, precious 1 hour of yours. Okay, so thank you the entire team of LJ University for giving me this opportunity. Really thankful, wonderful audience is there, full auditorium is full. I hope you are not sleeping. I'd also like to thank my organization also, LPU, who has given me the opportunity to talk over here. So I must thank my organization also to give me this opportunity. So thank you so much for being patient. Any question, I'm here.
Thank you, sir. The session is open for question and answers. Any questions?
Hello. My first question is how the bacteria concentration means it is bacteria eats the polysaccharide and drug is released. So how the concentration, like probiotic, how you give this much concentration is required?
Yeah.
So that is the first question. Second question, how you check the IV/IVC, in vitro correlation, because formulation in release in animal or human is different thing, and the concentration in nanogram is different thing, and the formulation in vitro it is micro mg per ml. So how you correlate that? That I want to know. So these
Yeah.
are the two basic questions.
Yeah, thank you. First question is how we quantify that how much bacteria we should take it, right? So your good name? Thank you. So this is very simple. We are not generating bacteria. The sterilized bacteria comes in a sachet. So the support we get from Unique Biotech in Hyderabad. Dr. M.S. Sudha is there. She is our also a collaborator or supplier of probiotics. She gives me ProMix and Biomix strain. Those strains are already standardized by them. That if you are adding 1 gram of the sachet, it contains this much CFU of the bacteria.
Yeah, that's yeah.
That is one thing. Second thing you can ask me, this is point of asking good question, how I have done for fecal microbiota. Because that also is bacteria. My objective was like we are taking from the market about the probiotic samples, right? Same way we tried to prepare with the fecal microbiota. So what we did that we have taken the 1 gram of the fecal matter and then we diluted it and we made it 1%. Out of 1% solution, we have taken in liquid form, okay, and then we have taken that weight also, and after that whatever the lyophilized mass came out, that lyophilized weight also we have taken, and based on that we sent it for metagenomic studies that this particular gram after lyophilization whatever the material has come, okay, this much gram is contained how much, because Microbiota is very tough to calculate, particularly when we are taking a randomized sample.
Yes.
Okay, so we have done the metagenomic studies. From metagenomic study, they have given us the data that 1 gram of bacteria contain flora is containing how much, and that process we did pre-lyophilization also and post-lyophilization also, because lyophilization time there are the chances of damage of the bacteria. So that also we validated. So that I have not shown, but if you see this my material today chemistry paper, which you have picked up, metagenomic results are there in that. Given. Second question, IVC. So already, if you are aware, I have talked about a biorelevant dissolution medium.
Yes.
So this is your question that in vivo lot of things will be there, particularly when for me it is not important that what is happening before
Yes.
upper GIT, because upper GIT में release ही नहीं आ रही है. Okay, in upper GIT it is not rating because kinetic pharmacokinetic is also showing in upper GIT it is not there. पहले हमें in vitro में fix करना पड़ता है. This is your question actually. So first 5 hours we will not add the any rat cecal content or probiotic based medium what we have created. Immediately at the end of 5th hour, means almost 4 hour 45 minutes, we will do the carbon dioxide purging. ओके ये मैंने नहीं बताया यहाँ क्योंकि मेरा नो हाउ है, टेक्नोलॉजी हिडन है. ओके बट हम में पेटेंट है तो कोई इशू नहीं है. ओके सो वी यूज टू डू द कंटिन्यूस पर्जिंग ऑफ कार्बन डाइऑक्साइड. विद द कार्बन डाइऑक्साइड इमीडिएटली वी यूज टू ट्रांसफर, इमीडिएटली हम लेते हैं उनका, क्योंकि द माइक्रोबायोटा विच इज प्रेजेंट इज नॉट ए स्ट्रिक्ट एनारोब, दे आर फैकल्टेटिव एनारोब्स. सो व्हाट वी डू दैट, we used to take out immediately in the medium. So before taking out the material also we'll purge it. The medium what we'll take 100 ml, we'll purge it rich in carbon dioxide, immediately लाके डाल देते हैं और after that 24 hour हो जाता है. So this is the way we maintain the in vivo in vitro biorelevant medium. क्योंकि IVC के लिए आपको biorelevant medium होना चाहिए, otherwise dissolution must become a bioavailable. Okay, thank you. Any other question?
Good afternoon, sir.
5 minutes है, 4 बजने में. I said I will not take more than 1 hour. Yeah.
Good afternoon, sir. My name is Manjot Singh. Pursuing Bachelor of Pharmacy from Sun3. Sir, my question is, जैसे कि आपने कहा कि जो drug release in colon delayed है या advance हो जाता है, I mean targeted में कितना different it would be?
आइडियली बेटा टारगेटेड करना है, एज आई सेड 90% तो जाना ही चाहिए. इफ यू आर गिविंग विद द 75% रिलीज, फिर तो सस्टेन रिलीज हो गया. 3/4 half lives of the formulation, if it has got released, okay, it will become a sustained release formulation. To make a full-fledged 4/4 of the quarter half lives of that, we need at least 90% formulation, at least 90 to 85 to 90% formulation should reach to colon. Then only you can say that it is a targeted delivery. पूरा चला जाए तो अच्छा है. अगर आप एंटी कैंसर ड्रग बना रहे हो टारगेटिंग बोले तो मेरे हिसाब से तो 0% होना चाहिए. नथिंग शुड गेट रिलीज बिफोर दैट. हमने देखा भी है 5% 6% ही रहता है. अगर आप अच्छे से पॉलीसेकेराइड की कोटिंग करो या पॉलीसेकेराइड, उसके लिए आपको मैट्रिक्स में पॉलीसेकेराइड डालना होगा और आपको कोटिंग भी करना होगा. एंड इट इज लिटिल चैलेंजिंग बट नॉट इम्पॉसिबल.
Thank you so much, sir.
Yeah, any other question? Thank you so much.
Thank you so much, sir. Now I request Dr. Sapna Malviya ma'am to felicitate sir with a memento. I request Dr. Sanjay Toshniwal sir and all the faculties to come up for a photo. After this, the next session will start, and this is an announcement that after completion of fourth session, the poster and oral presentation will start. This session is being chaired by Dr. Sachin Singh sir. I request Dr. Priya Shah ma'am and Shweta Gandhi ma'am to welcome our speaker with a tulsi pot.
Hi guys, welcome back to my channel. We are getting bored.
Thank you, ma'am. Dr. Sapna ma'am is professor and head of institute at Modern Institute of Pharmaceutical Sciences, Indore. She holds MPharm, MBA, PhD in pharmacy and a Doctor of Science in Pharmaceutical Sciences. With more than 17 years of experience in teaching, research and administration, she had made significant contributions to pharmaceutical education and research. She had published over 110 research papers, 17 books and 56 book chapters, and has guided numerous postgraduate research projects. Her academic leadership is reflected in her role as coordinator for NBA, AICTE, PCI, RGPV and other accreditation processes, as well as her contributions as a recognized examiner and resource person at national and international forums. She has 9 patents and 8 registered designs to her credit along with consultancy projects and funded research initiatives. Sapna Ma'am has been honored with several awards including the Academic Leadership Award in 2025, Distinguished Professor Award in 2022, Outstanding Women Educator Award in 2022 and Eminent Professor Award in 2021. She is also recognized as a ZED consultant by Quality Council of India and serves as State President of Women Forum Society of Pharmaceutical Sciences and Research. Her research interest focuses on phytopharmacology, drug discovery, diabetes, and plant-based therapeutics. With her dynamic involvement in professional bodies, editorial boards, and innovation forums, Ma'am continues to inspire excellence in pharmaceutical sciences and education. Now I request Dr. Sapna Malviya Ma'am to share some insights on the topic redefining drug development and AI-driven synergy and precision in pharma.
પાવર બેંક છે તો થોડું આગળ મોકલો
કેટલી 30 40 ગાઈડેક્સ કેસે તો ગાઈડેક્સ કાંઈ પણ બોલતો
So, very good afternoon to all my dear students, faculty, fraternity, respected dignitaries on the dais. See, LPJ University, I am from last one day, and I can say that you are at the wonderful place. Place is a beacon of excellence, and you have multidisciplinary courses over there. So, it's honor for me to be invited for the session at this university, and I would like to credit some of the things in this dialogue. So thank you very much, sir, Vice Chancellor, sir, the entire team, Almi ma'am, Shweta ma'am, everyone sitting over here to have me over here. So coming straight to the topic, I'll talking about redefining drug discovery and development, AI-driven synergy and precision in pharma. Actually, in pharma, when we talk about, we have a certain sort of myth that drug discovery is a time-consuming process. Obviously it is, but drug discovery deals with life and health of the people. And when we are talking about life and health of the people, security of the people, safety of the people, it's a matter of life and death. So it's not about the time. So here I am talking about the redefining drug discovery and development, wherein I am telling you that whatever AI is doing, with the integration of AI, will add synergy, precision, quality, sustainability, all the aspects regarding to it to make a safe, reliable dosage form. It is a drug discovery redefining with optimized time. Indian pharmaceutical industry is facing a lot of issues due to increased R&D cost. R&D cost is there. We cannot meet the challenges of the diseases. Approximately, when I am reading the literature for you, there are more than 30 metabolic disorders which are in the pipeline of development. So there are a lot of challenges driven in the path of drug discovery and development. Stating few of the experts which I was listening by this, the CEO of Insights is a company. He has mentioned that drug discovery and development, the process is optimized with the help of AI. Because AI has added precision, consistency, synergy in this aspect. So here we'll talk about drug discovery and development. Moving to the next slide. This is the disclaimer of my side that this talk I am sharing for academic and education purpose. here, these are the opening thoughts of my presentation, that in this presentation, we'll talk about a basic aspect because it's for students. We'll talk about what is drug, what is medicine, why drug and development is required, what are the traditional aspects, how redefining of drug development is there, what is artificial intelligence, what are the parameters over there, AI synergizing drug development, AI precising it, top pharmaceutical companies, what AI parameters this pharmaceutical companies are using, because I just want to state that, what is CEO has also stated that in one of the interview when he was asked that after 5 years where you are perceiving the pharmaceutical industry with the integration of AI. He said that after 5 years, I will not say that AI will be totally integrated in all the formulation, but then yes, various phases like target selection, validation, clinical trials, some phase of it can be speeded up by looking to the aspect. So what is the AI implementation in industries? Some case studies, how they are implementing it, and what is the role of pharmaceutical educators and students in this synergy. Because there are a lot of opportunities where we can develop ourselves with the integration of artificial intelligence. Yes, obviously some jobs will be lost, but then human power is very, very exclusively important for integration of this artificial intelligence in Industrial Revolution 5.0. So basic term, drug and medicine. If you see the word, there is 4 letters in drug and 8 letters in medicine. Obviously, we sometimes use it as a part of a synergy that they look like, but the purpose is entirely different. Drug means any chemical entity or API, and medicine is a 8-letter word carrying its potential for the therapeutic activity. Whenever you talk about the drug is the basic entity, but whenever you talk about the medicine, it is for therapeutic potential. So AI is not only going to develop new formulation, but AI will synergize and precise these new drugs into a suitable formulation which is safe, reliable and adaptable for all the patient's healthcare. These are the classification of drug and medicines. You will see. So I am just taking you through this journey of drug and medicines because it is highly correlated till end. Till end, there is a lot of correlation. I'll just share one very interesting insight how we developed the penicillin. If we go through the literature, penicillin was developed in a lab where they have just opened a petri dish and kept over there. When the scientists returned and came to the place, he saw that something is stopping the molds to grow. And there was the development of the penicillin. So our traditional methods of development of drug are hit and trial methods. These are hit and trial methods, but now due to the process is optimized to an extent and it takes a lot of correlated things over there. So we need to move with AI, and AI is predicting with the models. I'll show you few important videos which can add more understanding and inspiration about drug discovery and development. You can see that one molecule is classified in 4 different ways. In the similar way, medicine also, it is classified in 4 different ways. Now you can imagine these all molecules are being bifurcated in millions of directions and they have different therapeutic activity, and these all are interlinked with each other in different aspects. So to finding out the right target and validating it, going to the hit, getting the lead optimization is extremely difficult. I don't say that it is not possible, it is possible because we are sitting here and we are combating various metabolic disorders, but then the process is tedious and it is costly also. So AI is integrating identification part so smoothly so that it can be easy. Coming to drug discovery and development, I'll tell you 4 simple stages. It is a part of 4D. Drug, which I already told you what is drug, then discovery, development, and for disease. So these 4 Ds are total correlation of my presentation, which will be moving consecutively so that you can understand it more possibly. Drug, I told you, discovery of new things with the innovation, new formulation, either from existing one or new protein, something to be targeted with the disease. Then coming to the development part, development of formulation, considerations and pre-formulation aspects we will discuss. We'll touch the little bit so that you can get a little idea about the traditional approaches and new approaches which we are going through it. And finally the disease. Whole story is behind the disease because we need treatment approach for the disease. So drug discovery and development is a cycle which is simultaneously moving around so that you can get a proper formulation, targeted formulation for the specific site of the body and where it is drug is maximally bioavailable at the targeted site. So it's not an overnight process. Still it is not an overnight process. We are integrating AI, we are shortening our process, but still the time is there where we are anticipating over that we will reach very soon. So you can see that I just shared the example of paracetamol. It is a very common. I have many disorders in my mind, but simply you understand by invention of paracetamol. The major cause is pain and fever. Major cause is pain and fever. Targeted protein which is responsible or for causing it is prostaglandin, COX-2 inhibitor. Now this prostaglandin we need something to change the structure of prostaglandin or to inhibit the activity of prostaglandin or to reduce it in an extent, then it gave birth to the paracetamol. So this is a journey. Target can be a gene or protein. I'll show you. So it is not an easy task. I have taken a sample size of 10,000 molecules. Here target is something. We'll come to the slide later on. See, target is something which is either a drug or medicine. A target is something which is a protein gene which is causing the disease. So first step for drug development, you can see you need to understand the condition, what is the problem over there. Once you identify that target, next work you need to do is the validation of this target. Now this validation of target is very important because you need to understand which protein structure, which gene structure you need to change so that you can get an appropriate result where is exactly a problem. You can see in the video that you need to go with different meticulous operations over there for in target identification and target validation over there. Then comes hit. Hit is a molecule, it can be multiple molecule where you select the lead. Lead identification is there and this lead need to be optimized, and after optimizing it we go for formulation development, preclinical studies, clinical studies, and thereby new drug development and approval is there. So this is the first step, target identification and validation. If you can see over there in the drug discovery, you need to identify the biological targets over there, and it is a so meticulous process over there that the receptor, enzyme and nucleic acids are involved in the disease process. So you need to identify it and you need to find out that small molecule which can be converted to be druggable, or this molecule we need to rectify it by providing a suitable solution over it. This is a hit. Hit is a process, is an activity, a biological target. It has the measurable activity. Now this hit, we go through physical high throughput screening. We go through it physical. So there is millions of proteins are there, millions of parameters are there which we need to conduct. It goes with hit and trial, which is really tedious. If you are in the right direction in the lab, continuously working over there, then you can find out the aspect what we are doing. But now with the help of AI, you can go with virtual high throughput screening. This virtual high throughput screening adds the invention, adds the input of softwares and computers which you can easily identify the hit. Just revise for you that you need to first go for target identification. Target is nothing, it is a disease. If you understand in the raw language, it is a disease. Anything which is causing disease, it can be protein, it can be gene, that is a target. After getting the target, you need to validate the target that whether you have point out right target or not. After getting the target, you need to go for hit, understanding the hit molecule that have the activity for biological target. This is a process where hit molecule is converted into the lead molecule. Now these are aspects we are doing continuously in the lab. Now in the journey of continuity for transferring hit to lead, I'll just share one thing more with you. See, right now, AI is integrating in such a way that we have converted that solutions which were 50 years old. I'd just like to mention the software AlphaFold. AlphaFold has the potential to underline the 3D structure of the protein, because proteins are the real causes or real problems over there for the disease. Easily, if we identify the 3D structure of the protein, then we can fix a hit over it, and hit molecule by continuation and validation you can find a lead molecule, because there is a difference between hit molecule and lead molecule for achieving efficacy and understanding the different parameters. So this hit molecule and lead molecule, lead has the better pharmacokinetics, better understanding of the aspect in this. Here is the approaches in virtual high throughput screening, molecular docking. It's a lock and key hypothesis. You need to find a right molecule. You need to find a right key for the lock. You need to have that key at the proper position, and without jerk, the key should move continuously. That particular process of finding the chemical compounds go through molecular docking. By pharmacomodeling, pharmacophore modeling, you can go with the 3D structure and identification. So here we reach to the stage where you are there with the lead identification. So in COVID, we have observed the importance of AI. When we developed vaccine in few months, which usually take 10 to 15 years. So what the scientists have done, they have used AI over there to identify the protein, to crack the protein, to find out the right molecule over there. So Moderna is a company which has used this aspect to develop the COVID vaccine. Major metabolic disorders which a company is targeting. See, basically what happens whenever the company is selecting the product, they just find out the major metabolic disorder where the treatment is yet to be updated. Say it can be diabetes, because India is a diabetic capital of the world. It is a cancer where they are continuously working over there. Now you can see how there is a lead optimization, and how this lead optimization is done by very common methods, that is structure activity relationship. A small change in the structure can modify the whole aspect of that structure. And if we modify the whole aspect of the structure, its livability, its solubility, all the aspects are being changed. If these aspects are being continuously changing, then that affects the